So today we're going to continue our discussion of organ systems to talk about the respiratory system and the circulatory system. So for the respiratory system, we'll first talk about functions of the respiratory system. Of course, we have gas exchange, but there are some other functions that you may not be so familiar with, such as pH control, thermoregulation, and particle filtration. Then we'll talk about organs and the structural composition of this organ system. We'll discuss the mechanisms of breathing. And then we'll talk about the alveoli. So how do the alveoli enable gas exchange in the lungs? And then we'll talk about the circulatory system, or more specifically, the cardiovascular system. So the circulatory system is what's listed on the AAMC guide for this content category. But the circulatory system technically is consisted of the cardiovascular system plus the lymphatic system, but we're going to save the lymphatic system for a later lecture. So with regards to the cardiovascular system, we'll talk about functions of this organ system, the role it plays in thermoregulation, and then we'll just talk about circulation through our body. So what are the two different circuits of blood? And then what's the structure of the heart, and how does blood flow through the heart? Then we'll talk about endothelial cells, which line the vessels and organs of this system. We'll talk about blood vessels, so for example, arteries, veins, arterioles, capillaries, etc. And we'll discuss the structure of these vessels and key differences between them. And then finally, we'll talk about blood pressure, so the definition of blood pressure, maintenance of blood pressure, so the feedback mechanisms that enable it to stay at a homeostatic level, and regulation of blood pressure. And then we'll talk a little more about capillaries and capillary microcirculation. So, we know that the respiratory system is necessary for gas exchange. So our cells conduct aerobic respiration, meaning they need oxygen in order to produce the majority of ATP. So of course we have glycolysis, which doesn't need oxygen, but that doesn't produce much ATP. We rely on the TCA cycle and oxidative phosphorylation, and these require oxygen. We also know that a byproduct of these metabolic processes is CO2. So our cells in the tissue of our body need to constantly take in oxygen and get rid of CO2. And the respiratory system enables this. So specifically, oxygen is inhaled into the lungs, and then it goes all the way to the alveoli, which are little sacs in the lungs. And from the alveoli, O2 diffuses into the capillary beds that surround the alveoli. Then it travels in the blood until it's dropped off to the tissues of our body. CO2 diffuses out of the tissues of our body into the blood, travels to the heart, and then travels from the capillary beds surrounding the alveoli into the alveoli and is exhaled out of our lungs. The respiratory system is also crucial in thermoregulation, and more so for furry animals than less furry animals. But for most animals, breathing enables us to lose excess heat, so we breathe out warm and humid air. Furry animals like dogs are limited in their ability to sweat because they simply don't have the same surface area that enables sweating. And sweating is crucial for thermoregulation because it enables evaporative heat loss. So humans' main mechanism of losing excess heat is sweating. But for furry animals like dogs, they don't have that ability. And so these furry animals largely rely on the respiratory system to cool themselves down. So you may see dogs panting a lot more in the heat. And that's because breathing through evaporative heat loss enables them to get rid of that excess heat. So what is evaporative heat loss? Well, that's when evaporation occurs from the moist surfaces of either the skin or the respiratory system. And by breathing out that warm, moist air, they're getting rid of excess heat. And then finally, protection. So particulate matter is filtered out via the organs and tissues of the respiratory system. So when we breathe in, that air is not always clean. It's often filled with small dust particles and even microorganisms that could be pathogenic. So there are two main mechanisms that our respiratory system employs to get rid of this particulate matter. So first there's the first line defense, which is nostril hairs and mucus. Mucus lines the respiratory tissues up until the alveoli. And mucus and those nostril hairs trap small particles to try and prevent them from getting into the body. So also cilia line the airway, and cilia is going to sweep that mucus layer along to be swallowed and digested in the stomach, where that particulate matter can be digested. Alveoli lack mucus and cilia, so if a particle gets to the alveoli, what happens is the alveoli have their own resident macrophages, and these macrophages line the alveoli and locate, attack, and digest any particulate matter that makes it into an alveolus. Then the final important role of the respiratory system is pH regulation. So I should start by saying carbon dioxide is carried in the blood in three ways. Some carbon dioxide can travel freely in the plasma. Some is actually carried in the red blood cells as carbon dioxide. But the vast majority of carbon dioxide becomes carbonic acid or bicarbonate and is carried in this aqueous form. So in the blood, as I said, most of the CO2 combines with water to form carbonic acid. And that reaction is catalyzed by the enzyme carbonic anhydrase, which can actually catalyze the forward and reverse reaction. Carbonic acid is a weak acid. So as acids do, it dissociates to form its conjugate base, which is bicarbonate, plus a hydrogen ion. So you see we have two conjugates here, carbonic acid and bicarbonate, and that is what a buffer is. So as long as we have a sufficient amount of both conjugates, then we have a good buffer, which can resist changes in pH. So how does it do that? Well, if there's a sudden increase in protons in the blood, then bicarbonate can combine with some of those protons to form carbonic acid and resist a drop in pH from that influx of protons. Conversely, if there's a sudden influx of hydroxide ions into the blood, then some of the carbonic acid can donate a proton to those hydroxide ions to form water and to resist an increase in pH from that influx of hydroxide. So this blood buffer system is great, but sometimes it's set a little off balance. So let's talk about that. So blood pH has to be kept within a very tight range, and you should know these numbers. It could be helpful for the MCAT. So it's between 7.35 and 7.45. If the pH drops below 7.35 or above 7.45, it's called respiratory acidosis or respiratory alkalosis. And the respiratory, in those terms, means that the cause of the change in pH is either too little or too much breathing. So respiratory acidosis is when the pH drops below 7.35. Specifically, this is when excess carbon dioxide builds up, and that is going to cause a drop in the blood pH because we know that most of that CO2 combines with water to form carbonic acid, and carbonic acid dissociates and it donates a proton into the blood, and those protons are going to drop the pH. So if we think about Le Chatelier's principle, if there's too much CO2, that's going to combine with water to form carbonic acid. Carbonic acid is going to then dissociate even more into a proton plus bicarbonate. And so if we have too much CO2, we're going to push the reaction mechanism forward and end up having too many protons, which results in acidosis. So we know that this is when there's excess CO2. And there's excess CO2 when you're not breathing enough. So if you're hypoventilating, meaning you're not breathing enough, then CO2 is accumulating because you're not getting rid of it, because you're not exhaling enough. That drop in blood pH is going to be recognized in the pons and medulla by these special chemoreceptors. And that signal is going to result in an increase in your breathing rate. So that's the compensatory mechanism. If there's a drop in the blood pH, then your brain is going to tell your body to increase the breathing rate. And that is in order to get rid of that excess CO2, in order to raise the pH back to its homeostatic level. There's also renal compensation. So this was respiratory compensation, but renal compensation is carried out by the kidneys. So if the acidosis is bad enough, then the kidneys are going to excrete excess carbonic acid and reabsorb more bicarbonate. So how would that help? Well, we have to look at Le Chatelier's principle to understand that. So if there's acidosis, there's a drop in the pH. If you get rid of carbonic acid, then you're going to push this reaction more to the left. And if you push this reaction to the left by either dropping the amount of carbonic acid or increasing the amount of bicarbonate, so both of those push that reaction to the left, then we're going to form more carbonic acid by combining those excess protons with bicarbonate. So we're going to soak up some of those excess protons in order to raise the blood pH. And then there's respiratory alkalosis. So anything that's alkaline is basic. So this occurs when the blood pH is raised above the homeostatic level. And specifically, that occurs when there's reduced levels of carbon dioxide in the blood. So if there's less carbon dioxide in the blood, then less carbonic acid is formed. And if less carbonic acid is formed, so less of this, then this reaction equilibrium is going to shift to the left, right? Because if we drop the amount of carbonic acid, then the equilibrium needs to compensate for that by shifting to the left and forming more carbonic acid. And in doing that, so when this reaction mechanism shifts to the left, many of these protons are going to combine with bicarbonate to form carbonic acid. And if we get rid of those protons by combining them with bicarbonate, then we're going to increase the pH. So this is caused by hyperventilation. So if you're breathing too much, then you're getting rid of too much CO2. And again, the chemoreceptors in the brain are going to recognize this change in the pH, and they are going to send signals to your body to decrease the breathing rate. So this is caused by hyperventilation, but the compensatory mechanism would be to hypoventilate. Now let's talk about the structure and the organs of this organ system. So first, the nasal cavity. The nasal cavity filters, warms, and humidifies incoming air. Also, the hairs in the nostril and the mucus layer is going to trap particulate matter. Next, we have the trachea, sometimes called the windpipe. And this brings air that came in from the nose into the lungs. So this is the passageway from the nasal cavity to the lungs. The trachea is strong and it's reinforced by rings of cartilage. The trachea is going to branch into two bronchi, the left and right bronchi, which you can kind of see here. Then, of course, we have the lungs, which are the main organ of gas exchange. And the bronchi are going to repeatedly branch into even smaller tubes called bronchioles. And bronchioles terminate in an alveolus. So we can see here, the trachea is going to branch into these two bronchi, and the bronchi branch into bronchioles. And then at the end of bronchioles are these sacs of alveoli. So there's a specific type of bronchiole called a respiratory bronchiole. And this is a bronchiole that has some alveoli along its walls. And that means that the bronchiole can directly participate in gas exchange because the alveoli are where gas exchange occurs. And then we have the alveoli. So these are the sacs where the gas exchange actually occurs. So oxygen diffuses from the air in the alveoli into the blood. So as I said, there are capillaries that surround the alveoli. And oxygen is going to diffuse from these sacs into the capillaries, and CO2 is going to diffuse from the blood in the capillaries into the alveoli. And the alveoli have walls that are one cell layer thick. So that's crucial for the speed of diffusion. So you can see here, these are the capillaries, and they directly surround the alveolar wall. And the alveolar wall is so thin that the gas exchange between the blood in the capillaries and the space in the alveoli is very rapid. Finally, we have the diaphragm. So the diaphragm is this large muscle here, and this facilitates breathing. So it changes the volume of the thoracic cavity in order to enable inhalation, which is breathing in, and exhalation, which is breathing out. So now let's talk about the mechanism of breathing. So first there's inspiration, also called inhalation, and that's breathing in. So during inspiration, the diaphragm contracts, and when it contracts, it pulls downward. And the intercostal muscles, which are here in your chest, are going to contract, which pulls the ribs outward. And both of these are going to expand the thoracic cavity. So as the diaphragm moves down and the ribs pull outwards, then your thoracic cavity increases in volume. And as the volume of the thoracic cavity increases, the pressure inside the cavity decreases. So we know that P and V are inversely related in the ideal gas law. So as the volume of the cavity increases, the pressure decreases. And that causes air from the outside to rush into the lungs. Now this is called negative pressure breathing. And that's because the pressure inside the lungs is negative relative to the outside environment. So it's just less than the pressure of the outside environment, which is of course 1 atm. And air naturally moves into areas of lower pressure. So if your lungs or your thoracic cavity is lower pressure than the outside environment, and you open your mouth, then air is naturally going to rush into your thoracic cavity. And there are some terms that are on the AAMC content guide, so I want to make sure to touch on them. And one of those is differential pressure. And this term simply means the difference in pressure between two regions. There's also positive pressure breathing, and that's if, for example, during mouth-to-mouth CPR, when someone forces or blows air into your mouth, that would be positive pressure breathing. But the normal mechanism of breathing is a negative pressure mechanism. And now let's talk about exhalation. So in exhalation, the diaphragm relaxes, and when it relaxes, it actually moves upward. The intercostal muscles also relax, so the thoracic cavity is going to decrease. And as the volume of the thoracic cavity goes down, the pressure inside the cavity increases above the external environment pressure. And so air is going to rush out of the lungs into the outside environment. Now let's talk about the elastic recoil of the lungs. So after you inhale, the lung naturally rebounds. So after inhalation, exhalation naturally follows. And that's because of a number of different reasons. So after you breathe in, you can breathe in now, exhalation is very natural. You don't have to, of course it's an involuntary mechanism, but also the lungs naturally want to recoil back down to this expiration state after inhalation. So once you expand the thoracic cavity, there's this natural tendency of the thoracic cavity to decrease back down. And that's because there's connective tissue in the lungs that has elastin fibers. So that kind of, like when you stretch a rubber band, it naturally wants to pull back. Also, there's surface tension of the fluid that lines the alveoli. So surface tension is tension within a film of liquid that tends to pull the liquid molecules together. So it tends to decrease the surface area of a film of liquid. So surface tension of the alveolar fluid also facilitates this natural rebounding. Emphymema, and also called COPD, which is chronic obstructive pulmonary disease, are problems of breathing in which the elastic recoil is decreased. So in emphymema, the problem is with exhalation. So exhalation is not as efficient because the alveoli are damaged, usually caused by smoking. And that means that you can't get rid of that old air in the lungs as well. So you can't get as much oxygen-rich air into the lungs. Now let's talk about intrapleural pressure. So I did say that the lungs are surrounded by two pleura, which are thin membranes. So there are two pleural membranes that surround the lungs. And between those two membranes is a region called the pleural cavity, which isn't really a cavity, but instead it's a thin film of fluid. And so those two pleura are stuck to each other. So the film of fluid between them is like a glue. And why are they stuck to each other? Well, one of the pleura is attached to the lungs, so it surrounds and encases the lungs. And one is attached to the walls of the thoracic cavity. The pressure between those two membranes is usually less than the atmospheric pressure. So again, this negative pressure mechanism keeps these two pleura stuck to each other. And so that means that the one pleura that surrounds the lungs is stuck to the pleura that surrounds the thoracic cavity. So this keeps the lungs inflated, sort of, if you think about it in that way. It keeps the lungs from collapsing inwards, as they naturally would due to this elastic recoil. If one of the pleura is punctured, so for example by a stab wound or a gunshot, then you destroy this negative intrapleural pressure, and one of the lungs is going to collapse. And finally, let's talk about surfactant. So this is a substance that functions to decrease the surface tension of a liquid. So surface tension, as I said, tends to decrease the surface area of a film of liquid. And we know that there's a film of fluid that lines the alveoli. And if there was no surfactant, then that surface tension, which is very strong, would really cause those alveoli to collapse or completely decrease in size, so much so that gas exchange wouldn't be as efficient. So we need surfactant to decrease the surface tension slightly. And that prevents the alveoli from collapsing during exhalation. So we know that there's elastic recoil, but elastic recoil can't be too strong, otherwise the lungs would collapse. So this too strong elastic recoil or collapse of the lungs is prevented by this negative intrapleural pressure, which keeps the lungs sort of stuck to the thoracic cavity walls, and by surfactant, which keeps surface tension at bay. So this is a illustration of the pleural membranes. So you see we have these two pleura, one of them directly encases the lungs, and one of them is attached to the walls of the thoracic cavity. And the intrapleural cavity isn't really a cavity, instead it's a film of liquid that acts like a glue. And that's because the intrapleural cavity is negative pressure relative to the outside. So these two pleura are stuck to each other, which keeps the lungs linked indirectly to the walls of the thoracic cavity, which are the ribs and the diaphragm. So the information on this slide is not mentioned at all on the AAMC content guide, but I thought I'd include it because I've seen it mentioned in a number of different practice passages and practice tests. So a spirometer is a relatively simple and cheap instrument that's used to measure the volume of air inhaled and exhaled by the lungs. And specifically, it can measure a bunch of different volumes that can detect different functions of the lungs to make sure that they're functioning properly. And so all of these different measures do not need to at all be memorized by you, because when I did see this on the MCAT, usually the chart was provided or the information was given in some way or another. But it would be helpful to pause the video and look over this chart so that you're familiar with it. So, for example, a spirometer could measure total lung capacity, which is the total volume of air in the lungs after the maximum, so forced inhalation. Or functional residual capacity, this is the volume of air left in the lungs after a normal exhalation, versus residual volume, which is the volume of air in the lungs after a maximum exhalation. So after you maximally exhale, so a forceful exhalation, there's still some volume in the lungs, volume of air in the lungs. Or total lung capacity, which is the maximum volume of air that the lungs can hold. So you can see all of these different measures may be altered slightly if an individual has some breathing problem. So different breathing problems such as emphysema, COPD, asthma, they're associated with specific changes in these measures, and that's how it would come up on the MCAT. So I would just read over this chart just to, have seen it once and be familiar with it in case it comes up. Now let's talk about gas exchange in the alveoli. So capillaries are the site of gas exchange in the body. So in the lungs, the capillaries are going to drop off CO2 and pick up O2. And in the tissues, the capillaries are going to drop off O2 to the tissues and pick up CO2 from the tissues. And there are capillary networks that surround the alveoli. So how does this gas exchange work? Well, we've kind of touched on it, and it's facilitated by diffusion. So we know that molecules naturally move into an area of lower concentration. So in the capillaries that surround the alveoli, we have deoxygenated blood that's returning from the tissues to the lungs. And that blood is going to have a high concentration of CO2, which it picked up from the tissues, and a low concentration of O2 because it dropped off oxygen to the tissues. And so that blood, which has a high amount of CO2 and a low amount of O2, is going to naturally pick up O2 because oxygen is high in the alveolus. So O2 will naturally move into the blood. And CO2 is naturally going to move from the blood into the alveolus. So this exchange is, it's natural. It's the tendency of a molecule to move into an area of lower concentration. So overall in this exchange, CO2 moves out of the capillaries into the alveoli to be exhaled, and the inhaled O2 moves from the alveoli into the capillaries. Then in the tissues, the O2 is going to move from the blood into the tissues, and CO2 is going to move from the tissues into the blood. And then just one thing to mention, in these diagrams, when blood is colored blue, that means it has a low amount of O2, and when it's colored red, that means it has a high amount of oxygen. So Henry's law is one thing mentioned on the AAMC content guide, and for what you guys need to know, it's pretty simple. Henry's law is basically the amount of gas. Henry's law says that the amount of gas that can be dissolved in a liquid is directly proportional to two things: the partial pressure of that gas and the solubility of that gas. So when we talk about the respiratory system, Henry's law says basically that the amount of O2 that can dissolve into the blood is directly proportional to the partial pressure of that gas. So the amount of O2 that can dissolve into the blood is directly proportional to the amount of O2 that's in the alveolus. So if there's more oxygen that's in the alveoli, then more oxygen can dissolve into the capillaries. So if we have a breathing problem like emphysema, where exhalation is damaged, so there's a bunch of old air sitting in the alveoli, that old air is going to take up space and decrease the amount of oxygen that can be in the alveoli. And if there's less oxygen in the alveoli, then less oxygen is going to dissolve into the capillaries, and that's problematic. So you can see here, this is a diagram that's depicting the partial pressures of oxygen and CO2, and it can sort of help you understand the natural tendency of these gases. So in the air, there's a very high amount of oxygen and a low amount of CO2. Once we get into the lungs, and this is like depicting one alveolus, because there's some residual volume sitting in the lungs, the amount of oxygen is slightly less than in the outside environment, and there's slightly more CO2. But still, compared to the blood that's coming into the alveolus, this air has more oxygen and less CO2 than the blood. And then because of that, the oxygen is going to move into the blood, and the CO2 is going to move from the blood into the alveolus. And that gas exchange is going to continue until the partial pressure of oxygen and CO2 are approximately equivalent in the alveolus and in the blood that's leaving the respiratory system. Now let's segue to talking about the circulatory system, or specifically the cardiovascular system. So some functions of the circulatory system. 1, deliver oxygen to the tissues. So as we've said, oxygen that's been inhaled into the alveoli is going to diffuse into the capillaries and then travel through the vessels of the body to be delivered to the tissues. And we know that oxygen is required for cellular respiration. Nutrients, hormones, solutes, fluids, and any particle that's traveling in the blood can be picked up by the tissues as needed. So these can diffuse out of the capillary into the tissues down their gradient, or these solutes could be picked up via active transport. So, for example, the circulatory system is the method by which hormones reach their target organ. Carbon dioxide must be removed from the tissues and carried in the blood to be exhaled. The transport of carbon dioxide as carbonic acid and bicarbonate also serves the function of facilitating pH homeostasis. And then also metabolic wastes, so especially nitrogenous waste such as urea, travels in the blood to reach the kidneys. And the kidneys facilitate the excretion of these wastes in urine. So there are so many different functions of the circulatory system. And this great chart is a good summary of them. So not that you will need to memorize them. Many of them will be very obvious to you, but I would suggest pausing the video and just reading some of these roles of the circulatory system. And then this is a sort of general layout of the circulatory system. So we have deoxygenated blood returning from the capillary beds of the body into the heart. That deoxygenated blood is pumped from the heart to the lungs, and then oxygenated blood travels from the lungs into the heart, where it's pumped at high pressure through the rest of the body. So the lower body and the upper body. And as I said, in any diagram, blue is going to denote deoxygenated blood carrying a high amount of carbon dioxide, whereas red is low in carbon dioxide but high in oxygen. And then we should touch a little bit on thermoregulation. So the role of the circulatory system in thermoregulation. So there are vessels that are close to the surface of your skin that can constrict or dilate in order to regulate heat loss. So when you're cold, the vessels near the surface of your skin are going to constrict. So that's called vasoconstriction, when a vessel constricts. And that is in order to avoid heat loss. So when they constrict, less blood is going to travel through them. So less blood will be able to dissipate that excess heat. And then vasodilation enables more blood to travel through those superficial vessels, enabling more loss of heat. And heat is going to be lost through radiation and convection. So now we're going to talk about some terms before we dive into the anatomy of the cardiovascular system. And these terms, some of them are listed on the WMC content guide, and some of them aren't, but all of them you should know when you're learning about the cardiovascular system. So pulmonary circulation is the circulation of blood from the heart through the lungs to pick up oxygen and drop off CO2, and then back to the heart. Whereas systemic circulation is the transport of blood through the entire body and brain to drop off oxygen and pick up CO2 and wastes. So blood passes through the pulmonary circulation, and that's just one capillary bed that engorges the alveoli. Then it's going to go back to the heart to be pumped through the systemic circulation. So if we're following one red blood cell, that red blood cell is going to pass through one capillary bed in the pulmonary circulation, and then it's going to pass through one capillary bed in the systemic circulation. There is one exception to that, and that is if the red blood cell travels through a portal system. So a portal system is two consecutive capillary beds. So normally there's just one, but in a portal system there's two consecutive capillary beds, and this is important in the liver, so the hepatic portal system. And we talked about the hypothalamus pituitary portal system. So if blood travels through a portal system, then it travels through two capillary beds in the systemic circulation and one in the pulmonary circulation. Perfusion is a term that refers to the flow of blood through a tissue. Ischemia is when there's not enough blood flow through a tissue. And if there's not enough blood flow through a tissue, then that tissue is going to be starved of oxygen and nutrients, and dangerous wastes and metabolites are going to build up. So ischemic stroke is the most common type of stroke. So there's both ischemic stroke and hemorrhagic stroke. Hemorrhagic stroke occurs when a vessel bursts, and that blood is going to build up at high pressure in the brain, and that causes damage. But almost 80% of strokes are ischemic strokes, and that occurs when there's a clot in a vessel leading to the brain, and so a certain region of the brain is going to be starved of oxygen and nutrients. Then hypoxia is a term that refers to lack of oxygen in a tissue. So hypoxia would follow ischemia, of course. And then angiogenesis. This is the formation of new blood vessels. So this, of course, occurs during development, but it also occurs unnaturally or unhealthily during tumor growth. So when a tumor is growing, you see angiogenesis. The tumors are growing their own blood vessels in order to get a direct supply of oxygen and nutrients. Now let's talk about the heart. So the heart has 4 chambers. It has the right atrium and the right ventricle, and the left atrium and the left ventricle. Now when you're looking at a heart, a diagram of the heart, the right-left directionality is going to be reversed. That's because if you're looking at a heart as compared to the heart being inside of you, you can understand how the directionality is flipped. So this diagram has some structures that you don't need to know, but a lot of these you do need to know. So let's talk about the flow of blood through the heart. So in general, the right atrium collects deoxygenated blood returning from the body. So deoxygenated blood returns from the body in the vena cava, which is the largest vein in the body. So the superior vena cava is going to collect blood from the upper body, whereas the inferior vena cava is going to collect blood from the lower body. So that blood rushes into the right atrium, and it flows from the right atrium into the right ventricle. Now the difference between atria and ventricles is that ventricles are thicker and more muscular. So they are the pumps that send blood either through the pulmonary circulation, if we're talking about the right ventricle, or the systemic circulation, if we're talking about the left ventricle. So blood flows into the right ventricle, and then it's sent from the right ventricle to the lungs to exchange gases. So then it travels to the lungs, exchanges gases, and returns via pulmonary veins to the left atrium. Then it travels from the left atrium into the left ventricle, and from the left ventricle, it's pumped at very high pressure through the aorta, and the aorta branches into many different arteries that travel through the body. So when we talk about atria versus ventricles, ventricles, as I said, are the pumps of the heart. So although both of them do pumps, the atria has to pump in order to force blood from the atria into the ventricle. The ventricles have the job of pumping the blood through the entire body and through the lungs. So they're very muscular and have thick walls. Because of that, the pressure in the ventricles is very high. Also, when you're looking at the two ventricles, the left ventricle is actually thicker and more muscular than the right ventricle, and that's because the left ventricle has the tough job of pumping blood at high enough pressure so that it can travel throughout the entire body. So the left ventricle has to make sure that blood is traveling fast enough and at high enough pressure to reach all the way up to our brain and all the way down to our toes and our far out extremities. There are some main vessels that you should know. So I mentioned the vena cava, and these bring deoxygenated blood returning from the upper and lower body into the right atrium. The pulmonary artery brings blood from the right ventricle to the lungs. The pulmonary veins bring blood from the lungs into the left atrium. And the aorta brings blood from the left ventricle, to the rest of the body. So arteries pump blood away from the heart, and veins bring blood back to the heart. Normally, arteries are carrying oxygenated blood. But one exception is the pulmonary artery, and that's bringing deoxygenated blood from the right ventricle to the lungs to pick up oxygen. And whereas veins normally carry deoxygenated blood, an exception is the pulmonary veins, which bring oxygenated blood from the lungs into the left atrium. Let's talk about the valves of the heart. So here they're listed on the diagram. You can see there's a valve between the atria and ventricles. These are called atrioventricular valves. And then there's also valves right before the pulmonary artery and right before the aorta. The tricuspid valve is the valve between the right atrium and the right ventricle. So that's here. And valves are like doors that when they're closed, they prevent the flow of blood. And when they're open, of course, they allow the flow of blood. So the tricuspid valve only opens when blood flows from the atrium into the ventricles. The pulmonary valve is between the right ventricle and the pulmonary artery. So that's this valve here. So this valve is only open right after the ventricle pumps blood. And when the ventricle pumps, so the ventricle is going to contract, the blood in it is going to be at very high pressure, which is going to cause this valve to open and force the blood through the pulmonary artery to the lungs. Then we have the bicuspid valve, sometimes called the mitral valve, between the left atrium and the left ventricle, which of course only opens when blood comes from the atrium into the ventricle. And then we have the aortic valve. And the aortic valve is between the left ventricle and the aorta. So this opens when the left ventricle contracts. The blood in the left ventricle is at very high pressure, which forces open the aortic valve, and the blood travels through the aorta to the rest of the body. So we were talking about the blood supply to the body, but what about the blood supply to the heart? Well, the heart has its own blood supply, of course. And the arteries that supply the heart with oxygen are called coronary arteries. So coronary arteries branch from the aorta, and they supply the cardiac muscle with blood. And then coronary veins bring blood back into the right atrium. So then it's going to go from the right atrium to the right ventricle, through the pulmonary circulation, return to the left atrium, left ventricle, through the aorta, and then an early branch is going to be the coronary arteries, which supply the cardiac muscle. A heart attack is technically called a myocardial infarction. And this is just ischemia in the heart. So if one of these coronary arteries is blocked, then blood isn't going to get to a region of the heart, and the cardiac muscle is going to begin to die. Let's talk about systole and diastole. Systole is the pumping of the heart. So systole is when the ventricles contract, to push blood into the lungs and into the aorta to go to the rest of the body. Blood from the vena cava and the pulmonary veins enters the atria at this time, and in late systole, the atria contract to force the blood into the ventricles. Diastole is when the heart refills with blood after the ventricles have contracted. So this is like the resting period of the heart. I remember this because die, as in to die, is at the beginning of diastole, and so this is like the resting period, right? And the systole is the pumping of the heart. So this is just a summary of the flow of blood through the heart. If you want to pause the video and go through it, but this is definitely something that you should know before taking your MCAT. Let's talk about cardiac conduction. So the muscle cells of the heart are called a functional syncytia. And that's because they're all interconnected through gap junctions. Specifically, between the cells of the heart are these regions called intercalated discs. And the intercalated discs are a bunch of gap junctions that directly connect the cytoplasms of adjacent cardiac muscle cells. What's the purpose of this? Well, directly connecting the cytoplasms of these cells enables rapid spread of an action potential so that all the cardiac cells of a certain region are synchronized. Because we need all of the cardiac muscle cells of, for example, the right atria to contract at the same time. So cells of the right and left atria are connected, but they're separate from the ventricles because we need the atria to contract offset from the ventricles, right? The atria contract, then the ventricles, then the atria, then the ventricles. Cardiac myocytes, which are the muscle cells of the heart, have two types of voltage-gated channels: fast sodium channels and slow calcium channels. The slow voltage-gated calcium channels result in an action potential lasting longer than usual, which is crucial for the rate of our heart. Now, we know that most muscles contract when they get an action potential sent from a neuron. That neuron releases neurotransmitter onto the muscle cell. But the heart is different. The heart is innervated by neurons, which can increase or decrease the heart rate. But the heart has special cells that can actually initiate the action potential. So these cells are clustered into nodes. So a node is a specialized region of cardiac muscle cells that can generate an electrical impulse. So they act like both a neuron and a myocyte. There are two nodes that you need to know: the sinoatrial node, sometimes abbreviated the SA node, and the AV node. The SA node is the pacemaker of the heart. So this is a region of cells that's found within the right atrium that spontaneously and regularly depolarizes, causing the atria to contract. And the signal that's generated by the SA node spreads to the next node. And then the next node is the AV node, which is going to cause the ventricles to contract. So this is called the pacemaker of the heart because it sets the rhythm. Immediately after one action potential in the cells of the SA node, another one follows right after. And the action potential of the SA node is a little bit different from the action potential you're used to. But essentially, there's a huge amount of sodium leak channels in the cells of the SA node. Sodium leak channels are going to naturally let sodium rush into the cell, which is going to increase the membrane potential. That increase in the membrane potential, if it reaches the threshold, is going to cause the release of calcium from the sarcoplasmic reticulum, and then calcium is going to enable that huge spike in the action potential. So you don't really need to know in detail the action potential of the cells of the SA node, but you should know that it's the pacemaker of the heart. It spontaneously and regularly depolarizes, and it has a huge amount of sodium leak channels. Then we have the AV node. So the AV node propagates an action potential to the ventricles, specifically via the bundle of His and the Purkinje fibers. So let's look at this diagram here. Here we have the SA node that's located in the right atrium. A signal that's generated in the SA node is going to cause the atria to contract. Then there's a slight delay, and the signal travels to the AV node, which, as you can see, is located between the atrium and the ventricles. The signal travels to the AV node and then is sent via the bundle of His, which branches into these Purkinje fibers. And they are located throughout the ventricles. And so when a signal reaches the AV node, it travels through the ventricles, causing the ventricles to contract. So as I said, that signal from the SA node is delayed slightly so that ventricle contraction follows atria contraction. So let's follow that pathway of cardiac conduction one more time. So the action potential starts in the SA node over here in the right atrium. The SA node has those sodium leak channels. Sodium leak channels depolarize the cells of the SA node, and then that increase in the membrane potential causes these calcium channels to open, and calcium rushes into the cell, causing the spike in the action potential. We know that all of the cardiac myocytes of the atria are connected via intercalated discs, so the action potential spreads throughout the atria and the atria contract. After a slight delay, the signal reaches the AV node. The AV node is going to conduct that impulse through the bundle of His and into the Purkinje fibers, causing the ventricles to then contract. So you can see that here we have the SA node, atria contract, travels to AV node, and through the fibers to the entirety of the ventricles, and the ventricles contract. And it's also depicted in this diagram here. So signal at the SA node causes atria to contract, reaches the AV node, and travels through this bundle of His and Purkinje fibers to the walls of the ventricles, which then contract. Immediately following ventricular contraction, the SA nodes spontaneously depolarize again, and the cycle continues. So we know that the nodes generate the action potentials, but the heart is still innervated, and the innervations of the heart can increase or decrease the heart rate and the force of the heart contraction. So the heart is under control of the sympathetic and parasympathetic nervous system. So we know that the parasympathetic nervous system is the rest and digest system. So that's going to slow down the heart, whereas the sympathetic nervous system is going to increase heart rate and contractility. So the parasympathetic nervous system, so you can see that's called the vagus nerve. The parasympathetic nervous system controls the heart via acetylcholine. Specifically, acetylcholine delays the SA node to slow down its firing. So it's controlling the SA node in order to control the heart rate. And the action of the parasympathetic nervous system is always occurring. So the parasympathetic nervous system is always keeping the heart rate at bay, at this resting heart rate of 60 to 70 beats per minute, unless we need activation of the sympathetic nervous system. So without the parasympathetic nervous system, so without that vagal tone, the heart rate would always be at its maximum value, which would be extremely taxing on the heart. So instead, we have this vagal tone that's constantly occurring unless there's activation of the sympathetic nervous system. So if there's activation of the sympathetic nervous system under a fight or flight situation, epinephrine and norepinephrine are going to increase the heart rate and force of contraction. So although the heart does and can generate its own impulses, it's still under control of the nervous system. Now let's talk about endothelial cells, which line the entire circulatory system. So the endothelium lines the heart and all the blood vessels, and it's comprised of simple squamous cells. So remember, we talked about different structures of endothelial tissue. So there's simple and stratified, and there's squamous and cuboidal. And if you remember, the squamous cells are sort of like tiles on your kitchen floor, and simple means that it's one layer thick. So this is a single layer sheet of flat cells. The endothelial cells are not just a lining of the vessels, but they're also active players in vasodilation and vasoconstriction, inflammation, and blood clotting. So they perform some very crucial functions. So the endothelium produces many factors, such as nitric oxide, that regulate the contraction of smooth muscle cells surrounding blood vessels. So they can actively participate in regulating the diameter of these vessels. During inflammation, the endothelium allows white blood cells to adhere and enter the tissue. And the endothelium also regulates thrombosis, which is blood clotting, by preventing thrombosis when it's unnecessary and promoting it when it's necessary. The endothelium is a semi-permeable barrier, so it allows certain things through. It can become excessively leaky during extreme and prolonged inflammation. And if it's excessively leaky, then fluid is going to flow from the blood into the tissues, and that's when edema occurs. So you can see here is a diagram of the endothelium, and there's many junctions that allow certain proteins, water, and other solutes through. And even between cells of the endothelium, solutes and other molecules are allowed through. The endothelium is, as I said, not just a lining, but performs these crucial functions. So the endothelium is damaged in conditions such as diabetes and atherosclerosis, and smoking also damages the endothelium. And when the endothelium is damaged, a number of problems arise because, as you can see, it's crucial in healthy functioning of the circulatory system. Now let's talk about blood vessels. So blood flows from arteries, so from the heart into arteries. Arteries branch into smaller vessels called arterioles. Arterioles branch into even smaller vessels called capillaries, and that's where the gas and solute exchange occurs. Capillaries combine into venules, which are just smaller versions of veins, and venules combine into veins, and then veins return to the heart. So this occurs in both the pulmonary and systemic circulation, the only difference being in the systemic circulation, arteries carry oxygenated blood, and in the pulmonary circulation, arteries carry deoxygenated blood. So normally we're talking about systemic circulation, and that means that arteries are carrying oxygenated blood away from the heart. An artery consists of endothelium, smooth muscle, and connective tissue. Arteries are very elastic because they have to withstand very high pressure and large pressure fluctuations. So if you think of the aorta, which is the largest vessel, the aorta is carrying blood that was just pumped very forcefully from the left ventricle. And the aorta is the largest artery and has to withstand the highest pressure. All other arteries branch from the aorta. Arterioles are just smaller versions of an artery, so an artery branches into arterioles. Arterioles have a very similar structure to arteries, but they're highly involved in vasoconstriction. So they control the blood flow into capillary beds through contraction of smooth muscle rings. So precapillary sphincters are rings of smooth muscle that when they contract, they decrease the flow of blood into the capillary beds that follow arterioles. Capillaries are the vessels where solute and gas exchange occurs. They're the smallest diameter vessels and the thinnest, so they're a single layer thick. And they become so small in diameter that oftentimes they only allow red blood cells to travel in a single file line. Capillaries have no smooth muscle, so they cannot vasodilate or vasoconstrict themselves, but they rely on the contraction of smooth muscle in arteries or arterioles in order to control the blood flow through them. Capillaries coalesce into venules. As I said, venules are just smaller versions of a vein. And venules then merge into veins. So veins in the systemic circulation carry deoxygenated blood back to the heart. As I said, the exception is in the pulmonary circulation, and that's when the pulmonary vein brings oxygenated blood from the lungs back to the heart. Veins consist of endothelium, smooth muscle, and connective tissue. Veins have valves to prevent the backflow of blood. So in general, veins have valves, but arteries do not. And that's because arteries are carrying blood at very high pressure. So it's moving at high pressure and rapidly, so it's unlikely to flow backwards. But veins are carrying blood at low pressure, so they need these valves to prevent backflow. Also, smooth muscle and surrounding skeletal muscles contract to aid in the return of blood from the veins back to the heart. So here's a diagram of arteries, arterioles, capillaries, venules, and veins. And you can see the structure is quite similar. So they both have smooth muscle and an endothelium. There are a few different membranes that arteries have that veins don't. And some of the differences between them we've mentioned. So only veins have valves. Arteries are actually more muscular and therefore thicker. So you can see that ring of smooth muscle in the artery is thicker than it is in the vein. And there's huge pressure differences. So blood is at much higher pressure in the artery than it is in the veins. And you can see that the cross-sectional area of each capillary is less than the cross-sectional area of each arteriole. But when you sum up the cross-sectional area of the capillaries, it's greater than the cross-sectional area of the arterioles. So if the cross-sectional area of the tubes in the capillary beds are greater, then that means the pressure is going to drop. So if you think back to physics, think about the flow of fluid through different pipes. If you have fluid flowing through a pipe with a larger cross-sectional area, it's going to be moving at lower pressure. So this is a really great diagram that you should definitely know for the MCAT. And it shows pressure differences in different vessels. So the aorta is where you're going to have the highest pressure. And you can see that these fluctuations are the systolic pressure, so when the heart is pumping, and the diastolic pressure. And the pressure is pretty high in the aorta and all arteries. Then it drops when it gets to arterioles, drops dramatically when it gets to capillaries, and then decreases all the way to almost zero when it's back to the heart in the vena cava. Now let's talk about blood pressure. So we know that systole is when the heart is contracting. So systolic pressure is the pressure exerted by the heart, specifically contraction of the ventricles, on the vessels of the body as blood is being pumped through the body. And then diastolic pressure is the pressure in the vessels when the heart is momentarily resting. So we know that diastolic pressure is always lower than systolic pressure. So that's when we talk about blood pressure as 120/70, that's naming the systolic pressure over the diastolic pressure. So what is normal blood pressure? Well, normal blood pressure is pressure that's less than 120/80. And this is measured in the arteries. So you can see here, if this is depicting, your average person, you're measuring their blood pressure in the arteries of their arm, and you're getting a systolic pressure of around 120 and a diastolic pressure of around 75 or so. And if it's greater than this normal range, then you are in a hypertensive range, and hypertension is chronic high blood pressure. Now, this term pulse pressure, you should know, is a single number, which is calculated by doing the systolic pressure minus the diastolic pressure. So in an individual with a blood pressure of 120/80, the pulse pressure would, of course, be 40. And those numbers are in millimeters of mercury. So mmHg is millimeters mercury. Now, how is blood pressure increased? Well, blood pressure is increased in two ways. So if there's increased peripheral resistance to blood flow, or increased cardiac output, then you're going to have increased blood pressure. So let's talk about what these two are. So cardiac output, sometimes abbreviated CO, is the amount of blood flowing through your vessels per unit of time. So it's liters per minute, for example. Cardiac output can be calculated by doing stroke volume times heart rate. So if you look at the units, this makes a lot of sense. Cardiac output is liters per minute. Stroke volume is how much your heart pumps out per beat. So that's liters per beat. And then heart rate is beats per minute. So if you multiply stroke volume times heart rate, beats cancel out, and so you get liters per minute. So you should know these abbreviations and what these terms mean. So if cardiac output is increased, that can increase blood pressure. So we can increase cardiac output then by increasing heart rate, all other factors staying the same, or by increasing stroke volume. So if your heart is beating harder, for example, or beating faster. Increased peripheral resistance is caused by global vasoconstriction of arteries. So you know that smooth muscle in arteries or arterioles will contract. Or increased blood viscosity can also lead to increased peripheral resistance. And peripheral resistance is just, if you want to think about it, it's like resistance to the flow of blood. So vasoconstriction is going to decrease the diameter of the pipes. It's going to make it harder for blood to flow through those vessels. And then large-scale vasodilation is going to result in, of course, decreased blood pressure. Increased water retention by the kidneys. So, for example, the hormones ADH and aldosterone can trigger increased water retention. Is going to result in increased blood plasma volume, so increased blood volume. And increased blood volume converts to increased stroke volume most of the time, right? And so that will also result in increased blood pressure. So changes in blood pressure are, of course, pretty complex. So this is a summary and a big generalization, but you should know how all of these changes can affect blood pressure. The last thing we have to talk about is capillary microcirculation. So this is a capillary, and this is the arterial end, so it's closer to the arteries, and this is the venous end. And capillaries are, of course, bathed in interstitial fluid. Now, there's hydrostatic pressure in any vessel, and that's just due to the fact that blood is being pumped at high pressure by the heart through these vessels. So hydrostatic pressure, you can kind of think of it as blood pressure. And hydrostatic pressure is just the fact that the blood is being pumped at high pressure through these vessels, and that tends to force fluid out of the vessels. And specifically, it's going to force fluid out of the capillaries because fluid cannot leave the arteries or veins. And there's also osmotic pressure, which we've spoken about before. So remember, osmotic pressure is due to different solute concentrations on both sides of a semi-permeable membrane. and the endothelium of capillary beds is a semipermeable membrane. So blood is carrying a bunch of solutes and proteins, and it's hypertonic to the surrounding fluids because of that. And because it has a higher overall concentration of solutes, the surrounding fluid has a tendency to want to move into the capillaries by osmosis. So there's hydrostatic pressure, which has a tendency to force fluid out of the capillaries, and osmotic pressure, which has a tendency to bring fluid into the capillaries. Now, usually, as long as everything is healthy, there's a net balance achieved. So some fluid is forced out via hydrostatic pressure, and some comes back in via osmotic pressure, so that the overall volume of blood stays about the same. Nevertheless, at the arterial end of the capillary beds, the hydrostatic pressure is slightly higher than it is at the venous end. So osmotic pressure is constant throughout the capillary bed, but hydrostatic pressure is a little bit higher at the arterial end, and we can see that here. So in a capillary bed at the arterial end, the blood pressure is slightly higher. So the hydrostatic pressure is slightly higher at the arterial end of the capillaries. So that means that at the beginning of the capillary bed or at the arterial end, there's slightly more fluid pushed out, but then as we get towards the venous end, there's more fluid being brought in. And as we've said, if the hydrostatic pressure is very high, so in the case of chronic hypertension, and/or capillaries are leaky due to constant inflammation, then excess fluid is going to leak into the tissues, and that's edema. Okay, so that concludes this lecture on the respiratory and cardiovascular systems.